I'm Qindong Zhang, a master student from molecular medicine at NTNU. Welcome to my master thesis defense. DNA molecules are constantly damaged in our bodies. Among others, double-strand breaks are the most toxic DNA damage because they can result in the loss of a large DNA region. If left untreated, double-strand breaks can bring detrimental effects like immunodeficiency, neurodegenerative disorders, and increased susceptibility to cancer. At present, many people are suffering from such disorders, but current knowledge of specific proteins and their functions involved in DNA repair is incomplete. During my master thesis project, I'm focusing on the genetic interaction between two DNA repair factors, XLF and MRI. Double-strand breaks are induced by either exogenous or endogenous agents. Among endogenous agents, there are two physiological events. They are VDG recombination and class-switch recombination. Double-strand breaks are introduced in developing B and T cells during VDG recombination to assemble antigen receptor genes, contributing to a broad antigen receptor repertoire. In addition, double-strand breaks are generated in primary B cells upon antigen stimulation during class-switch recombination to produce antibodies of different isotypes. Non-homologous antigen, or NHEJ, is required to detect, process, and repel double-strand breaks generated during VDG recombination and class-switch recombination. Therefore, deficiency in NHEJ factors can result in immunodeficiency. In the presence of double-strand breaks, Cu70, Cu80 form a ring-shaped heterodimer Cu threads on the broken DNA ends, which in turn recruits and activates DNA PKCS, a protein kinase catalytic subunit, forming DNA PK holoenzyme with Cu. The formation of DNA PK complex further recruits endonuclease artemies to process the broken DNA ends. Eventually, the end ligation is completed by LAC4, XRCC4, XLF complex with the help of Cu. In addition, depending on the complexity of the broken DNA ends, several NHEJ accessory factors are recruited, such as PAX and MRI. PAX and MRI are two recently described NHEJ factors. Their roles in NHEJ still need further investigation. In 2006, several children and teenagers with profound severe combined immunodeficiency, microcephaly, and growth retardation were reported, where these patients have recombinant trait. A gene encoding a previously uncharacterized NHEJ factor was mutated. Later, this factor was named XLF or cynonoise. Unlike human, XLF deficient mice are almost normal and possess modest B and T lymphopenia. So, what is the reason that makes such a big difference between XLF deficient human and mice? This is due to the functional redundancy between XLF and other NHEJ factors and DNA damage repair factors. Moreover, the combined deficiency of XLF with other proteins have revealed their genetic relationships, like DNA PKCS, PAX, 53BP1, and H2AX. Thus, XLF deficiency in lymphocytes can reveal previously hidden NHEJ activities of other proteins. I decided to use the XLF deficiency in mice to investigate the genetic interaction between XLF and MRI in vivo. But here comes a problem. Inactivation of either XLF or MRI resulted in leaf-born mice. However, combined activation of XLF and MRI leads to embryonic lethality, It undoubtedly challenged the study of genetic interaction between XLF and MRI in vivo. So, what is the solution? Looking for the solution, I noticed that in Hong's paper published in 2018, they found massive neuronal apoptosis in the central nervous system of mice lacking XLF and MRI when compared to mice deficient in XLF and mice deficient in MRI. The neuronal apoptosis reminiscent of mice lacking LAC4, XRCC4, and mice with combined deficiency of XLF and PAX, as well as XLF and DNA PKCS. All these mice are embryonically lethal and possess increased levels of neuronal apoptosis in the central nervous system. Moreover, the neuronal death in most cases is correlated with p53-mediated cell signaling pathway. p53 is a pro-apoptotic factor that mediates cell cycle arrest and apoptosis. So, is it possible to inactivate p53 in order to let the neurons harboring repelled double-strand breaks prevent the apoptosis and to get livable in mice? The answer is yes. Previously, it has been demonstrated by my group members that inactivation of TRP53 rescues the synthetic lethality between XLF and PAX, and XLF and DNA PKCS, leading to livable in mice. But it was unclear whether inactivation of TRP53 rescues the embryonic lethality caused by combined activation of XLF and MRI. For this study, I propose that inactivation of TRP53 rescues the synthetic lethality between XLF and MRI. Second, mice lacking XLF and MRI exhibit a growth defect and possess a skeet phenotype. In this study, I found that inactivation of TRP53 rescues the embryonic lethality caused by combined activation of XLF and MRI, leading to a livable mouse model. This livable mouse model provides me with the opportunity to study the genetic interaction between XLF and MRI in vivo. As shown in this picture, this is only one XLF MRI double knockout mouse detected in the process of the analysis. It is smaller in size as compared to the age and gender matched MRI deficient mouse. As per the statistical analysis, 6 XLF MRI TRP53 triple deficient mice were analyzed. When compared to wild-type XLF and MRI single knockout controls, mice lacking XLF and MRI have reduced body weight. Besides, the thymus isolated from mice lacking XLF and MRI is smaller in size, as shown in this picture. Combined deficiency of XLF and MRI resulted in reduced thymus weight and drastically reduced counts of thymocytes when compared to wild-type XLF and MRI single knockout controls. As shown in this picture, the spleen isolated from a mouse lacking XLF and MRI is obviously smaller than the one isolated from an MRI deficient mouse. When compared to wild-type XLF and MRI single knockout controls, mice lacking XLF and MRI have reduced spleen weight and reduced counts of splenocytes. To characterize the development of B and T cells, I chose flow cytometry. This is because the developing B and T cells display a sequential cell surface markers. Therefore, the developing B and T cells at particular stages can be characterized by their cell surface markers. For example, in the bone marrow, the cells displaying beta20 and CD43 but without IgM, are characterized as pro-B cells. I established and optimized a protocol for flow cytometric analysis. Particularly, I was the first in my group to optimize a bone marrow staining. In the process of the experiment, first, I took out bone marrow, thymus, spleen from the mice. Second, I obtained single-cell suspension by smashing the organs. Third, the cells were stained with fluorescent antibodies. After incubation, the cells were analyzed by flow cytometry. Bone marrow is a place where B cell development occurs. In the bone marrow, combined deficiency of XLF and MRI resulted in increased proportion of pro-B cells in comparison to wild-type XLF and MRI single knockout controls. Moreover, as compared to wild-type XLF and MRI deficient mice lacking XLF and MRI possessed reduced proportion of IgM positive B cells. In summary, mice lacking XLF and MRI have defective B cell development, and the B cell development is blocked at early stage in mice lacking XLF and MRI. Thymus is a place where T cells develop. In the thymus, mice lacking XLF and MRI possess drastically reduced counts of double positive T cells, CD4 positive T cells, and CD8 positive T cells in comparison to wild-type XLF and MRI single knockout controls. Together, T cell development is affected in the absence of XLF and MRI. Here, the upper left box marks CD19's positive splenic B cells. The lower right box marks CD3 positive splenic T cells. When compared to wild-type XLF and MRI single knockout controls, mice lacking XLF and MRI possess drastically reduced counts of CD19 positive B cells. Although the reduction is significant, the fractions of CD19 positive B cells in mice lacking XLF and MRI is clear. Likewise, compared to wild-type XLF and MRI single knockout mice lacking XLF and MRI have reduced counts of CD3 positive T cells. The reduction of CD3 positive T cells is significant, but the fractions is clear in mice lacking XLF and MRI. Therefore, there are small but clear fractions of splenic B and T cells in mice lacking XLF and MRI. When it comes to splenic T cell subsets, in this diagram, the upper left box marks CD4 positive splenic T cells. The lower right box marks CD8 positive T cells. When compared to wild-type XLF and MRI single knockout controls, mice lacking XLF and MRI possess drastically reduced counts of CD4 positive T cells and CD8 positive T cells. Although the reductions are significant, the fractions of both splenic T cell subsets in mice lacking XLF and MRI is clear. Together, there are small but clear fractions of splenic T cell subsets in mice lacking XLF and MRI. In this study, I also characterized the genetic interaction between MRI and PAX in vivo. I propose that MRI genetically interacts with PAX in vivo. To test this hypothesis, I applied the same methods that have been used for characterizing mice lacking XLF and MRI. I found that MRI PAX double knockout mice are comparable to MRI deficient mice. Their lymphoid, fertile, possess normal body weight and number of B and T lymphocytes. To study the impact of combined inactivation of MRI and PAX on class-free recombination efficiency, I established and optimized a protocol for class-free recombination assay. In the process of the experiment, I took out the spleen from the mice. The primary B cells were then isolated using magnetic beads. The isolated cells were then stimulated with lipopolysaccharide and interleukin-4 to induce class switch from IgM to IgG1. After cell culture for 72 and 96 hours, the cells were harvested and stained with fluorescent antibodies. After incubation, the cells were analyzed by flow cytometry. In this histogram, class switch to IgG1 were significantly reduced in MRI PAX double deficient B cells when compared to wild-type control at both 72 and 96 hours. However, there was no significant difference between MRI deficient and MRI PAX double deficient B cells with respect to the percentage of IgG1 positive B cells at both 72 and 96 hours. Although I don't have PAX deficient mice, it has been demonstrated by my group members that primary PAX deficient B cells exhibit normal class switch recombination levels. Together, MRI is required for efficient class switch recombination, while PAX is not. Based on the results, I drew the following conclusions. First, synthetic lethality between XLF and MRI is TRP53 dependent. Second, mice lacking XLF and MRI exhibit growth defect and possess a leaky skeet phenotype. Third, MRI does not genetically interact with PAX in vivo. In the end, I would like to thank for your attention, and I would also want to express my sincere thanks to my supervisor, Valentin, for his continuous supervision and guidance. I also want to thank my group members and colleagues, friends, and family for their support along the way.